All right, so I am going to take some time to go over the Unknown Organisms project with you so that you can be sure that you know exactly what it is that you need to do and what is expected of you for this project. So on Canvas, I have this document posted, so if you want to follow along, feel free to go ahead and print that out or take a look at it while we're going through this video. Typically, at this point in this semester, what we would be working on is our Unknown Organisms project, which is basically you are given an unknown organism and you have to identify it over the course of the next several weeks. So the way that you would do this is by utilizing your lab notebook that you've been developing throughout the course of the semester. You would have only your lab notebook available to you in the lab room while you're doing your lab work in order to identify your unknown. So I have this write-up, and I have it written up the way that I would typically, for the most part, have it written up for if we were meeting in class, if we were meeting in person. And that's so that you can kind of understand the project still and what we're getting at. So perhaps when we're talking about this project, come at it with the thought that we would actually be in the classroom. You would actually be taking your organism and trying to determine what it is. So what we're going to do is we are going to still do this preparation assignment, which would be done normally in the semester. You would do this ahead of time so that you would have this prepared as you are in the laboratory trying to determine what your organism is. So at the beginning here, I just say in this project, you'll draw upon many of your new skills to identify an unknown bacteria. You'll be given a bacterial culture, asked to identify it to the furthest degree possible, which may be different for each student, but you should be able to get down to the genus species, with the goal of determining the genus and species. So the organism selected includes types that you've studied this semester. So if we were in the classroom, what would be available to you is everything that we've used in the classroom. So that's why it says also that you need to employ tests that were learned during the semester to characterize morphological, nutritional, physiological, and biochemical characteristics of your unknown. So basically you would be handed an unknown, and then throughout the course of several weeks you would have every single type of media and supply, including incubators and things like that, slides and microscopes available to you to identify this unknown. This preparation assignment is to prepare you to be able to identify the unknown so that when you walk into the classroom, you know exactly where you're going to start and where you're going to go. So this project, and of course we're not going to be in the classroom, just remember we're approaching this from if we were, so it makes a little bit more sense. So for this assignment, you will have 2 weeks to complete it. Please do not wait until the second week to start this. Even if you feel like you don't have time during this first week or this first couple of days, what I highly recommend that you do is that you at least start to look at it and kind of wrap your mind around how to do it and how you plan on doing it, because then at least you can kind of have an idea in your brain of how much time it's going to take up, okay? For some people, this takes up a huge amount of time because of the way that their brain works. For some people, it does not take up that much time, but it does still take a pretty good chunk of time. So be sure that you're aware of that and that you're taking the time to do this. So in order to receive full points for the assignment, you must fulfill each of these following that we're going to go through. So again, you have 2 weeks to do this from the day that I'm posting this, which means that it's due by Tuesday, April 28th by 1 p.m. So uploaded to Canvas on the assignment by 1 p.m. So the first step is what you're going to be doing is creating a diagram or a flow chart that's going to allow you to determine the identity of the unknown organism in the lab. It needs to be easy to understand. I have to be able to follow it, and also would be useful to you in your lab work. So if you were going into the laboratory and I handed you a test tube with bacteria in it, you would know the very first test you're going to run to try to at least start to figure out what it is. And then you would know each test after that you would be running based on your results from the previous test. So that's why I say a diagram or a flow chart, and I'm going to show you an example in a few minutes of one of those. So you need to somehow devise a plan to where if you were to walk into the laboratory, you know exactly what test you're going to start with, and if you get a positive for this test or a negative for this test, then you know what test you'll do next in order to further figure out what organism you have. So in the second part, your diagram should be limited to these following organisms. So these are all organisms that you have used throughout the semester. That's why they're on this list. So if you were going to be in the classroom and identifying an unknown organism, it would be one of these organisms. So your diagram or your flowchart must include being able to identify every single one of these organisms, because you wouldn't know which organism you were handed. So you have to figure out a way to separate out and divide up all of these organisms into a final test that you can say, yes, for sure, this is this organism. Step 3 is your diagram must use tests that we have run on organisms throughout the semester. You do not have to include all of the tests that we have run, but you must include only tests that we have and that you have run in the class. So this would be assuming we went through and we did all the labs up through 13 that you guys have written up in your lab notebook. We would be in the classroom, and again, you would only have the media that we have. So even if you were online and you were trying to research, B. megatherium, and you found out that you could do some test for B. megatherium that allows you to identify it. If it's not something that we had used in the classroom and therefore have the media for, then it's not a test that you can do. Also, the fewer tests that are needed to get a positive identification is the better. So imagine if you actually are in the laboratory. Another piece to this unknown assignment is to prepare you for perhaps working in a laboratory. So if you were in a laboratory where you were getting samples from patients and you need to identify what bacteria they have or virus, but if you knew it was a bacteria at this point and you were trying to identify what it was, you would not go into the laboratory and then just take every single test that is available to you, every test possible in the entire laboratory, and swab and swipe and deep and everything on every single test, because that would be an incredible waste of resources. So what you would do, however, is you would attempt to identify that bacteria with the fewest resources possible, so that money isn't spent, that money isn't going to the patient, etc. So you want to make sure that your diagram or your flowchart is utilizing the fewest number of tests. So with that, you must be able to identify all of these organisms in 5 tests or less. So keep that in mind as you're going through, that you want to try to use tests that are going to separate out these organisms as early as possible, so it takes you 5 or less tests. And that again is not only just because in a laboratory, like a clinical laboratory, for example, or even a research laboratory, they would want to minimize the use of the tools and therefore the costs. But also because if we were actually doing this in the laboratory, in the classroom, you would only have a certain amount of weeks to do this. We couldn't spend, the next 10 weeks allowing you to run every single test and determining the answer. So you have to do it in 5 tests or less. And the fewer, the better. 4, the assignment must be all your own work and in your own handwriting. So make sure that you have it all written up. If your work is plagiarism checked and any or all of it is plagiarized, you will receive 0 points for the unknown preparation assignment. So this is a portion of your total score for your unknowns. And I'm going to be posting information about what portion or how many points goes to each. This is typically 20% of your final score. I may adjust that slightly because of our current circumstances, but that is something that I'm still kind of working on, and I'll let you know as soon as I know. Step 5, the diagram can be written in your lab notebook or on a separate paper or separate papers. It can be in black and white, or you can color code it for ease of understanding. So if you were to go into the laboratory, I know many students in past semesters like to color code it so that when they use it, they can see it, and they know what their flow is, and they know what they're looking for, and it's easier. And that may be easier for you too, even though you won't be doing it in actual laboratory. But I do recommend that you do any of those things. Typically, I have students each semester write it in their lab notebook, because remember, in the unknown organisms assignment in the classroom, the only thing a student is allowed to have is their lab notebook and a pen. So it would need to be in the lab notebook. However, since we are not actually going into the laboratory with these things, and you'll be posting this online, it can be either written in your lab notebook, or you can do it on separate pieces of paper if you don't want to have the lines and things like that. So you can do it separately, and then you will submit this assignment on Canvas. I made a note in the announcement, but also here, please note that the actual assignment itself is going to have 0 points associated with it. And that's because I am going to be taking all of the unknown assignment and lab notebook points and putting it all together at the end for the 100 points that your lab notebook / unknown assignment is worth. So there's an additional line item there for the grade book that is the full 100 points that I will be placing scores in at the end of the semester once every piece of it is submitted. All right, so that kind of goes over the document here. I do want to show you an example. So I'm going to start with the zoomed out example here. This is an example from a student in the past. That's why you can kind of see my red marks and things like that. But I blacked everything out so that you could at least see what it is that I'm talking about when I talk about a diagram or a flow chart. So I did leave the first test that they chose up at the top here so that you can see that. I'm going to zoom in a little bit, or a lot. So you can see that what they chose to start with was the gram stain. So they said, okay, if I walk into a laboratory and I am handed a bacterial colony, whether it's on an agar plate or a deep or a broth, I'm going to take that, and the first thing I'm going to do is I'm going to perform a gram stain. Now, when I perform the gram stain, then what I'm going to do is I'm going to separate out the different organisms, and what it's going to tell me is whether or not it's gram positive or whether or not it's gram negative. And on your diagram, it must be separated like this. You must then take whatever is over here, so whatever you're separating. In this case, we're using the gram stain, so we're separating gram positive and gram negative. Here in the box, all of these blacked out spaces, these are all of the list of organisms that would then be over here on this side. This is the list of organisms that would be on this side. So in this case, for gram stain, these are all the gram positive organisms. These are all the gram negative organisms. So if you can imagine, if you walk into the laboratory, you have your bacteria, you do your gram stain, you say, okay, it's a gram positive. So now you know you have one of these organisms here. Or it's a gram negative. Now you know you have one of these organisms here. So you must, in your diagram, have whatever you're separating out, every single organism that would be the positive or the negative for that, or whatever the positive negative equivalent would be. And what I mean by that is then I have blacked the rest of this out so that you do the work on your own. But here, so we know that this list here were all of the organisms that this person thought was gram negative, or should have shown up gram negative. And then this arrow down is going to go to the next test that they would have done on those organisms to try to separate them out further. Now, the test is written here in this darker green region. And then whatever this test was, then it ending up one way, so let's say positive for whatever it is, would then give this list of organisms. Now, the negative, if that's what it is, would then give this organism. And all of the yellow highlights here are end organisms. So you can see that if the person indeed had a gram-positive organism and then they did this test by week 2, they would have done gram stain on week 1 and inoculated whatever this is here. By the second week, if it was negative, I'm pretending here, if it was negative, they knew that they had this organism. So every single organism needs to be able to be separated out individually. So you can see all of the yellow ones are individual organisms, so that after that test is complete, you can say, this is positive. If it's positive, it's this organism. Or this is negative. If it's negative, it's this organism. So again, these are the gram positives. She or he ran this test, and then whatever the answer was would give these organisms. The opposite answer would give this one. So then this person said, okay, if I now have these organisms, what test could I run to separate them further? So then they said, I'll run this test, and this test, it looks like, would give 3 different answers, perhaps. So if it gave this answer, then we knew that we had this organism. If it gave this answer, then we knew that it would have this organism. And the this answer part is what I have underneath the lighter green here. So under the lighter green, it would say, for example, positive or negative. I just didn't erase it up here in the gram stain area so you could see what I'm talking about. So, and then they said, okay, if it ends up looking this way, then it could be any of these organisms. And then continuing on. So out of these organisms, if it ends up looking like this, then it's going to be one of these. So if it's one of these, how can I then further break it down so that I can figure out which specific organism I have? So go down here and run this test. So if I run this test, if it ends up this way, I know I have this organism. If it ends up this way, then I have one of these organisms. And again, the next test, this way, this organism, this way, one of these organisms. All the way until there's this final test here, and that's splitting up the last two organisms, which means if it ends up this way, I have this one. If it ends up this way, I have this one. And so you can see on both the gram-positive and the gram-negative. So I do want, again, to note, you can see all of my red scribbles and red marks, which means that this person perhaps did not quite understand which ones go where and if the tests ended up that way. The reason I used this as the example is because they did a very nice job of separating it out so that I could very clearly show you exactly what I mean by what you're supposed to do. So keep that in mind, that don't try to fit into this mold of saying like, okay, if it's gram-negative, I should be able to do 1, 2, 3 tests and figure them all out, because that perhaps is not the case given all of these red marks that I put on here. So, and I don't even know what these were underneath here right now. So I can't even say with certainty how many it would take. But so just keep this in mind. This is how it should work. If it works to go vertical like this, then that's great. If it works for you to go more horizontal, then that's fine too. I just have to be able to understand it. I have to be able to follow your reasoning. So if this is the answer, I go here. If this is the answer, I go there. Some people are not able to put it all on one page. So what they'll do is they'll say gram stain, for example, and they'll say gram positive, gram negative, and then they'll say on gram negative, they'll put an asterisk here and say see page 72. And then if I flip to page 72 or scroll to page 72, then on page 72, I can see the breakdown of the gram negatives even further. Or on, gram positives and asterisks, and, it say see page 80. And then I go to page 80 and it's all separated out there. So if you need to use multiple pages, please feel free to do that as well if you need more space to do that. All right, so that is going to conclude this video. Again, you have 2 weeks to complete this. Please do start working on this as soon as possible so that you have plenty of time. And regardless of technical issues or anything, you have 2 full weeks to get this in and get it submitted. I highly recommend that once you do submit it, that you back out of Canvas, back out of your web browser, then go back in, sign back in, and check to make sure it's there. Not only should you check to make sure it's there, you should also click on it to make sure that it looks and includes everything you want it to include. So keep in mind, it does need to be one page, one single document, meaning not one page, but one single document with all of the information, so I can just scroll back and forth and follow all of your arrows and your path and know exactly where you're going with what you're doing. So if you have any questions, please do post them in the general questions discussion board, because likely your question will help others, and others might be able to answer it, and you could get some really good helpful information that way. So